So next we will see the types of culture. There are different types of cultures are there according to the type of plant tissue we take, like callus culture, suspension culture, organ culture, leaf or leaf primordial culture, shoot tip culture, complete flower culture, anther or pollen culture, ovule and embryo culture, and protoplast culture. So the callus culture, that is the important one. A tissue that develops in response to injury caused by physical or chemical means. More cell which will form and differentiated, which are differentiated, maybe and are often highly unorganized within the tissue. So in nature, this wound tissue produced after a wound is formed in a plant to form a protective layer of cells to cover an injury is known as the callus. But in culture medium, when the tissue or cells are placed in a media, it develops into, in a agar media, it develops into an unorganized mass of undifferentiated cells that are known as the callus, and it needs to be subcultured every 3 to 5 weeks. And um, this is the picture of the tobacco callus. Then cell suspension culture. Suspension culture is a type of culture. It is a suspension culture, not a solid culture. So tissue or cells are suspended in liquid media and is agitated at an intervals, or agitated all the time, so that the tissue or cells will be individual cells will be in suspension, and they call and these are called the suspension culture. They develop into new tissues and later plants. So this is the suspension culture. So it contains a uniform suspension of separate cells, because always it is in agitation mode. They will be shaken, which is in a liquid media. So liquid media agitated continuously, and finally the cell gets separated. So it won't form a mass like callus. The cells will be individually separated from each other, and we can subculture that from the suspension culture. And this is done by rotary shaker attached to an incubator at a rate of 50 to 150 rpm. And this is how these suspension culture is maintained. From the callus, the suspension culture is maintained, and this is agitated or shaken, and so the callus becomes single cells in the second passage. This is again subcultured into a new jar and cultivated. Next is protoplast culture. Culture means for the isolation of protoplast, we need to do some mechanical or chemical treatments. The cell without the cell wall, the plant cell usually have the cell wall in it. So the cell wall has been removed by mechanical or other chemical means or enzymatic process, and what we get after that is known as the protoplast. So protoplast is very easier to culture rather than the whole cell with the cell wall, and so it can be introduced into nutrient media, and can be cultured to form the callus. So this is the process of protoplast culture. We remove the leaves, cut the leaves, and incubate in the enzyme, so that the cell wall is being removed, and protoplast isolated, centrifuge, and protoplast is being taken, sedimented out, and they are cultivated. So for the isolation of protoplast, two methods are being selected, that is the mechanical method and enzymatic methods are there. And for the mechanical method, plant tissue is taken, cell is able to undergo plasmolysis, microscopic observation of the cells, and we mechanically remove, cutting the cell wall with a knife, and remove the cell wall and release the protoplasm. Then for the enzymatic method, we use some enzymes. So leaves are sterilized, remove the epidermis, plasmolysed cells treated with pectinase plus cellulose, and protoplasm released. And in another stepwise enzymatic treatment is done. First it is treated with pectinase, then treated with cellulase to form the protoplast. Then meristematic culture. Meristem is the tip of or highly dividing cells on the tip of root and shoot are known as the meristems. So these are highly reproducing cells, so we can easily cultivate it into a new plant. Usually shoot apical meristems are taken for meristem culture. So it produces virus-free germplasm and mass production of desirable genotypes can be obtained. Then anther culture or pollen culture, we isolate the anthers from unopened flower bud and cultivate it in a culture medium, that is known as anther culture, and usually it gives rise to haploid plant. So production of haploid plant and production of useful gametoclonal variation can be obtained. This is how anther culture is being done. Embryo culture. So young embryos are removed from the developing seeds and then it is cultured in culture media to form the seedlings. Ovary and ovule culture, we isolate the ovary from the female plant and we get a haploid form of unfertilized cells and it develops into new plant. Achievement of in vitro fertilization and haploid plants can be developed. And these are the different kinds of tissue culture according to the type as well as the type of tissue we take. So advantages of tissue culture are availability of raw materials, fluctuation in supplies and quantity. So availability of raw materials like some essential oils or other plant derived products can be produced in mass production using this tissue culture. Then fluctuation in supply, if there is any lack or lack of supply, we can actually get rid of that. Then patent rights are the political reasons, easy purification of the compounds like secondary metabolites can be easily purified. Then modification of chemical structure is possible. Disease free and desired proper yield can be maintained. Crop improvement, biosynthetic pathway of these secondary metabolites can be achieved, like natural products from plants can be obtained by this tissue culture. Then immobilization of the cell can be done. So these are the main application, like we can make virus free plants, apical meristem culture through this plant tissue culture. Then we can have or so we can produce alkaloids, saponins, steroids, anti-tumor agents, biopesticides, food additives, chemicals, etc. As the secondary metabolites or drugs, we can make in large amounts of drugs, plant derived drugs from this tissue culture. Then forestry crops can be obtained through tissue culture, like if some plant is endangered or it is very rare, it can be tissue culture. And fruit crops can be cultivated. Vegetations or vegetatively propagated plants can be cultivated. Then plantation crops, like any plantation, any teak trees or any other plantation can be obtained. Then essential oils can be yielded from these plants. So these are the main applications of plant tissue culture, and that's all about plant